So in today's class we will go step by step to the regulation of gene expression. We will try to understand how cells control their genes, and then in this guide we will explain, it will be explained how cells turn genes on or off. When should the gene be turned on and when should it be turned off, or whether it should be turned on or whether it should be turned off. So gene regulation, it enables development, it adaptation, and for health it is the enabler. So it is the foundation of cellular specialization and response. So what is gene expression? It essentially means that there is first genetic information flow. So these genes contain instructions which are encoded in DNA. So DNA contains nucleotides. They contain nucleotides and these are copied to RNA, and this RNA is then translated to proteins. Then the next part of gene expression is that there is selective activation. So not all genes function in every cell. Each cell type expresses a specific subset of genes. So they may contain all the cells, the liver or brain or heart, they may contain the same subset of genes, but not the same subset is expressed. Selective heart specific genes are expressed in heart, lung specific genes are expressed in lungs, kidney specific genes are expressed in kidney, so on and so forth. Not only that, some genes are expressed early in development, some genes are expressed late in development. So there is in early development some genes are shut off and some genes are shut on. The next is protein production. The ultimate goal is making functional proteins like enzymes, structural proteins, etc. They are used to perform cellular tasks. So why gene regulation needs to be gene expression needs to be regulated? First is environmental response. The cells must adapt to changing conditions. Temperature suddenly changes, so the cell, in order to survive, it has to have some subset of genes to be expressed which will help them during heat stress. So the gene regulation allows for quick responses to environmental signals. So example, cells activate stress response genes during heat shock. Next is the cell specialization. So we have hundreds of types of cells and each needs different proteins to function properly. Muscle cells express contractile proteins while neuron express neurotransmitter receptors. So contractile proteins are not needed in neurons, so they are shut off. But this is exactly the philosophy behind it. Cell specialization. Next is energy conservation. As we know that protein production, it requires significant energy. That is very essential. Lots of ATP is being utilized. So cells avoid making unnecessary proteins. So digestive enzymes are only made when food is present. So what are the key stages of gene regulation? First is signal detection. Cells sense environmental or internal cues. There is a cue or a hint that it should start now, environmental change. Then comes transcription regulation, where control of DNA to RNA that is regulated. Now express higher, now express lower, now don't express. So this is actually regulation at the level of transcription. Then is RNA processing. Once the messenger RNA is made, it is processed There are modifications to RNA structures. They can also act as regulators. Some make the mRNA more stable, so more protein is made. Some short-lived, less stable, so less protein of that is made, and that's perhaps what's required by the cell. The next is translation regulation, where protein synthesis is controlled. And finally, you have post-translational modification. Proteins are phosphorylated, ubiquitinated, depending which regulate their stability, whether these proteins after translation, whether they will be active or not. So the starting point is the signal detection. The signal may be an external signal, hormone or environmental factors arrive, heat stress, cold stress, or a particular hormone is being secreted, thyroxine for example. Next is receptor binding. The signal or the molecule, it attaches to the cell receptor. Then there is a signal cascade or signal transduction which takes place. Finally, specific genes activate or deactivate, that is the gene response. The next level of control is epigenetic regulation. Most important among them is DNA methylation. So CH3 group, they are chemical markers. They are added to the DNA. So what happens? Once when that particular base is methylated, it is not accessible for transcription. It blocks. These methylation patterns, they are inherited during cell division. The next thing is histone modifications. You know that histones pack the DNA, the huge amount of DNA which is present in the cell. It is packed. So when histones they are packed, they pack the cells. So if you phosphorylate or acetylate, this packing structure, it opens up and the DNA is accessible for activation. The genes are accessible for activation. And the chromatin structure, where loose chromatin, euchromatin, allows gene access, whereas tight chromatin, heterochromatin, restricts the access. These changes can persist throughout cell generations. Next is chromatin remodeling. You know, DNA is tightly wound around histones, and when it is wound around, it is not accessible to transcription factors. So there is a remodeling complex like NRF or Swiss-Snif complex. They use ATP energy and they shift the histone's position. So the region, the gene region, the DNA region is exposed now to transcription factors. And chromatin opening takes place. DNA loosens in specific regions and transcription machinery can easily access everything. So transcription is the main control point. First important player is the RNA polymerase. It's that enzyme that reads DNA and creates RNA copy, central to the transcription process. And the primary regulation is that most genes control happens at this stage. It is efficient and energy saving. Just hold on for a moment. Okay, I'm back. So most gene control happens at this stage, primary regulation, through transcription factors. So some transcription factors, there are promoters before genes, they bind and it is regulated when it should bind, when it should not bind, where it should bind, and say the on-off decisions where cells decide whether to make RNA from each gene. So there is the critical go or no go checkpoint here. So transcription factors, like I told, cells on off switches, they active the gene expression takes place when transcription factors bind to promoters of genes, and messenger RNA is made and active protein production takes place. There are activators which enhance the RNA polymerase binding and transcriptions. There are repressors, which these transcription factors, they can be repressors which block RNA polymerase binding. And there are finally the transcription factors that recognize specific DNA sequence, like heat shock transcription factors binds to heat stress elements on the promoters of genes. You have promoters and operators. You have the promoter region, that is the DNA sequence from after which the transcription begins. Then you have the RNA polymerase binding, the enzyme attached to start transcription, and operator region where repressors can block activity. So there are regions where the repressor, like LAC-I repressor, comes and binds and it stops transcription. You have enhancers and silencers. The enhancers may not be just upstream of the gene, they can be billions and billions of nucleotides away, very far away, but somehow they bend and twist and they bind to the enhancer binding sites and enhance or boost gene activity, or for the case of silencer, they decrease the activity. Both bend the DNA to influence the promoter. These elements work through DNA looping. So they essentially form something like this. The enhancers bind here, here is the RNA polymerase, and transcription starts in this region. The next step comes in mRNA processing, especially in eukaryotes. In eukaryote mRNA, there are certain processes which take place, that is 5' capping. Special cap added to the mRNA start. This protects from the mRNA from degradation and aids in translation. Then the next thing which takes place is splicing, where introns or non-coding regions, they are removed, or exons, coding regions, they are joined together. Then you have a poly A tail which is added. This increases the stability and aids in export from the nucleus to the cytoplasm, where it is going to be translated by ribosomes either on the endoplasmic reticulum or in the cytosol, depending upon which mRNA it is or which signal factor is present, signal recognition protein is present. The next is nuclear export. Mature mRNA is transported to cytoplasm and it is ready for translation to protein, where ribosomes will bind. Another thing which takes place is alternate splicing, which takes place. So it can be that one gene contains multiple exons or coding regions, and they have introns also in between. Now it can be that intron also can make a viable protein when these introns, all the introns are not spliced out similarly. Maybe the first intron remains, the second intron is spliced, and the second intron, when it's joined in conjunction, they can make a functional protein. So from one gene you can have alternate from different types of mRNAs being produced. Different exon combinations create unique mRNAs. Each can produce a distinct protein variant. And this alternate splicing, it greatly expands the protein repertoire. Humans produce over 100,000 proteins from 20,000 genes. How is it possible? From 20,000 genes, you're supposed to make one third of it, that is something like 6,000 proteins, 6,000 to 7,000 proteins, but you have over 100,000 proteins because of this alternative splicing. So one gene is making more than one type of protein. The next part is mRNA transport and stability. mRNA lifespan is an important part. mRNAs have limited lifespan, some last minutes, others days. The shorter they stay, less amount of protein is present. Longer they stay, more protein is produced. Then there are degraded factors. Specific sequences mRNA can target it for quick breakdown. There are some sequences present in the mRNA when they are present, they autocatalyze themselves and kill themselves. You have stability elements also similarly present, which extends its productive life. Some mRNAs are transported to specific cellular location before translation. So it can be located to the endoplasmic reticulum or to the cytosol, depending on the signal recognition peptide which is present. Signal sequence it's called. Now we come to the translation regulation. Translation has got some initiation factors which control the assembly of translational machinery. For example, you have read about the eukaryotic initiation factor 2, which get phosphorylated during stress. Then you have the ribosome binding sites, affect efficiency of ribosome attachment. So these ribosome binding sites help the ribosomes to be more stably attached to the messenger RNA. For example, the shiny Dalgarno sequence in bacteria, Pribnobox. Then you have in eukaryotes, you can say it's the Kozak sequences. Then you have the regulatory proteins. They bind mRNA to block translation. Some regulatory proteins, they block the mRNA. They bind to the mRNA, and so ribosome cannot bind. Example, iron regulatory proteins. Then you have small RNAs. They bind to mRNA to inhibit translation. MicroRNAs, which is very important. So the 60% protein variance is found, percentage of gene expression differences between cells due to translational control. So cell to cell there is 60% difference in the proteins which are being made because of translational controls. In a specific cell, a specific type of cell, a specific regulatory protein comes and binds and stops translation. There are over 2000 plus human miRNAs, microRNAs, which regulate translation in human cells are found. And there is 30% energy savings. Potential cellular energy is saved by blocking translation versus transcription. Now finally we come to post-translational modifications. First is phosphorylation, addition of phosphate groups. They can activate or deactivate proteins. You can glycosylate proteins, addition of sugar protein. It is important for protein folding and stability. Then you have proteolytic cleavage. So you cut proteins to activate them, common in digestive enzymes like trypsin comes from protrypsin. So chymotrypsin to trypsin, it breaks, you cut it. So a little peptide is cut and the trypsin becomes activated. Ubiquitination, that you add a ubiquitinyl tag and these proteins are targeted for degradation. Faster they are targeted for degradation, less the protein can act on the cell. In prokaryotes, there is no nucleus. Transcription and translation, they occur together. The regulation is simpler regulation. They work in terms of operon. Example, lack operon, groups of genes controlled together, and they respond faster to environmental changes. Eukaryotes, the nucleus separates the process, all transcription in the nucleus, translation in the cytoplasm. And ER, it is a complex multi-layered regulation. Extensive RNA processing takes place. There is a chromatin level control which we have seen, and there is more post-transcriptional regulation. So this gene regulation is very important in development and health. It helps in cell differentiation. They allow a single fertilized egg to develop into hundreds of cell types, because unique genes, they may have all the genes, but only a subset of relevant specific genes are expressed in one type of cell, and a different subset is expressed in a different type of cells. So though they are basically having the same genes, still they look different, they express different proteins. In development of disease, you can find misregulation of genes lead to disease like cancer. Tumor suppressors may be silent when oncogenes activate inappropriately. Then you have environmental adaptation. Gene regulation allows organisms to respond to changing conditions. So it enables adaptation to diverse environment. So to summarize, gene regulation is equal to the life's control panel. Proper regulation ensures cellular function. It ensures precise control when and how much protein is to be made. Regulation of DNA and RNA and protein levels. There are multiple control layers, and there is the gene activity control, and that is, there is mechanism to turn genes on and off. So this is my class today. Do you people have any questions? এটা নিয়ে questions আছে? Okay, so no question. হ্যাঁ
স্যার বলছিলাম কি পোস্ট ট্রান্সলেশনের সময় কি সব ইন্ট্রন্স গুলো কি কাট হয়ে যায়?
না, সব হয় না, সব সময় সব হয় না, অল্টারনেটিভ স্প্লাইসিংও হয়।
আচ্ছা।
ইন্ট্রনটা স্প্লাইসই হলো না, ওটা জুড়ে গেল, ওটাও ট্রান্সলেট হয়ে গেল, ওটাতে কোনো স্টপ কোডন এলো না, ওটা পুরো প্রোটিন তৈরি করে ফেললো। এটাই তো হয়, অল্টারনেট স্প্লাইসিং হয়। ড্রসোফিলা তোরা পড়েছি, সেক্স ডিটারমিনেশন ইন ড্রসোফিলা, ওই এসেক্স সেলটা মেলেতে আলাদা হয়, ফিমেলেতে আলাদা হয়, যেটা মাস্টার রেগুলেশন, সেক্স লিথল।
আচ্ছা স্যার,
ঠিক আছে, ওকে ওকে দেন, তাহলে বাই বাই।